these are a few examples, could be in the form of a toll-like receptor, and these are basically a very common receptor involved in signaling pattern recognition. Although there are a lot, but they probably have a slight different structure in terms of the domain. But in general, they have this kind of structure. So this kind of structure is known as toll-like receptor. So although there are a lot of them, the differentiation of this receptor usually is in terms of which type of molecule they recognize in the pathogens. And the way we differentiate them is basically based on number, just number 1, number 2, number 3. TLR2, for example, recognize peptidoglycan. TLR4 recognize LPS. So in terms of the component of recognition is different, they are numbered differently. Okay. So we finish with the first subtopic. So I will continue with the second subtopic, 7B. Do you have any question regarding this subtopic, guys? So this subtopic is a bit general, but you need to understand the concept of the recognition and the difference between innate and the adaptive. So we need to know how innate recognize the pathogen, what kind of cells would have this kind of ability, and example of the innate receptor. For example, they are divided into 2 types: endocytic, signaling, and you need to know a few example of molecule that this receptor recognize. Okay, so I assume you all don't have any question, so I will quickly continue with the second subtopic. So here I'll go also a bit quickly, because these are quite a common topic for all of us, which is phagocytosis. So we need to know that phagocytosis comes under innate immunity. So since we are very familiar with this topic, so what is actually a specific information that we need to know under this topic? So first, I'm sure you all still remember what is phagocytosis. It's a mechanism involving a process of ingesting. I think this is too small. Okay. Process of ingesting and digesting microorganism by an immune cells. So that is phagocytosis. So next thing we need to know that phagocytosis is basically a term referring to a general type it's a general term referring to a group type of cells. So any cell that can ingest, and digest microorganism is called phagocytes. But we have actually learned before this in the immune in topic 2, right? In the immune cells, there are different kind of cells, right? So the next question we need to ask is, so what kind of immune cells could be actually a phagocyte, can become phagocytes? So there are a lot of them, we also know. And among these different type of cells, the major, most active type of immune cell is neutrophils and macrophages that usually carry out active phagocytes. So they become phagocytes. So these are 4 basic phases of the phagocytosis. So although we can basically still remember on the phagocytosis process we've learned in the previous courses, right? But under here, we need to break down the step of the phagocytosis into 4 basic steps related to the immune system, immune response. So first, to perform phagocytosis, the cell need to have the need to initiate the process with chemotaxis. Okay, still remember what is chemotaxis? Taxi for the chemical, right? Chemotaxis. Can anyone define chemotaxis, guys? Can anyone volunteer to define chemotaxis? What is chemotaxis?
Movement of an organism or cell in response to chemical stimulus.
There is a very formal answer to define chemotaxis. Okay, good. Anyway, yes, correct. So it's a process of chemical attracting cells to mobilize towards certain direction, right? In another way of explanation. So we need to know that phagocytosis process is not a passive process, it's an active process. So it's not that when the immune cells suddenly unexpectedly come across pathogen. No, there should be an active stimulation by the uh, by the by the breach of immune defense. For example, the pathogen enter, so there will be a deliberate stimulation, then the immune cells will know and come over. So this is the process that we know as chemotaxis. So certain protein is released, then the macrophage will be aware. So that is the first step. So second step would be recognition and adherence. Recognition of adherence, what? The pathogens. And followed by engulfment, and lastly digestion. So we'll go quickly. So here is just to show you an example of protein usually act as an chemotactic factors. So we've discussed before this regarding topic 6 regarding cytokines, right? And if you can still remember, there are how many family of cytokines? How many group, family class of the cytokines? And one of them, the last one in the list, is chemokine, right? So chemokine is included as one of the chemotactic factors. So they could be a type of cytokine that stimulate chemotaxis. So there's also could be, these are also an example of a common one, C5A is a substance that is produced by complement pathway activation. So this also could be chemotactic factors. These are example of chemotactic factors. Is basically a chemokine here, a cytokine, the protein, the protein CXCL1 and 2 that is produced by neutrophils. Okay. So these are the detailed steps whereby you can see here the pathogens, bacteria, for example. These are the chemotactic factors. So when there is a presence of microorganism, certain nearby protein will cause C5A to be released, to be produced, and therefore it will become chemotactic factors for the phagocytes to uh, smell there is some infection occurring here. And these are the C3b